Brazilian wasp venom kills cancer cells, but not healthy cells. Wasps get their fair share of bad press. They have painful stingers, and they're not as useful or cute to us as bees. However, their time to step in the spotlight may be just around the corner. Their venom has been shown to attack cancer cells while leaving healthy cells alone. The cancer-targeting toxin in the wasp is called MP1, Polybia MP1, and until now, how it selectively eliminates cancer cells was unknown. According to new research, it exploits the atypical arrangement of fats, or lipids, in cancer cell membranes. Their abnormal distribution creates weak points where the toxin can interact with the lipids, which ultimately pokes gaping holes in the membrane. These are sufficiently large for essential molecules to start leaking out, like proteins, which the cell cannot function without. The wasp responsible for producing this toxin is the Polybia lister. The toxin has so far been tested on model membranes and examined using a broad range of imaging techniques. You can see the team's research results in the Biophysical Journal. Cancer therapies that attack the lipid composition of the cell membrane would be an entirely new class of anti-cancer drugs, said Paul Beals from the University of Leeds and co-author of the study. This could be useful in developing new combination therapies, where multiple drugs are used simultaneously to treat a cancer, by attacking different parts of the cancer cells at the same time. In healthy cell membranes, the inner layer facing the inside of the cell is packed with phospholipids, including PS, phosphatidylserine, and PE, phosphatidylphenylamine. However, in cancer cells, PS and PE are located on the outer layer of the cell membrane, facing the opposite way. To test the different effects of PS and PE's presence on a cell, the scientists examined how the MP1 interacted with model membranes infused with PE and/or PS. The presence of each phospholipid had a destructive effect on the cells. PS increased the chance of MP1 binding to the membrane by a factor of 7 to 8. The presence of PE inflated the size of the holes created by the MP1 by a factor 20 to 30. Formed in only seconds, these large pores are big enough to allow critical molecules such as NR and proteins to easily escape cells, said João Ruggiero Neto, from São Paulo State University, and co-author of the study. The dramatic enhancement of the permeabilization induced by the peptide in the presence of PE, and the dimensions of the pores in these membranes was surprising. The next stage for this research is to adjust the amino acid sequence of MP1, to see what gives it its selective properties, and to try and refine them. Understanding the mechanism of action of this peptide will help in translational studies to further assess the potential for this peptide to be used in medicine, Beals says. As it has been shown to be selective to cancer cells and non-toxic to normal cells in the lab, this peptide has the potential to be safe, but further work would be required to prove that.